Okay, so tonight's video is ideally going to we're going to be able to fit in different types of vessels, some information about blood pressure, as well as how nutrients and waste are transported like back and forth in and out of the bloodstream. If we can get through all of those things in this one video and keep it a reasonable length, then that is every bit of material that we will cover regarding vessels for this particular portion of the chapter. So we're going to base everything pretty much off of diagrams. So you'll want your diagrams handy while we're going through and we're looking at all of these different materials. So we're going to start first with this picture here, comparing our three basic types of vessels to one another. Okay, so we have artery as one type of vessel. And we've talked about this before. Our definition of an artery is that it is taking blood away. So get your A there, away from the heart. Okay. And then we have our vein, which is going to be taking blood to the heart. So you'll notice nowhere do we talk about oxygenated versus deoxygenated. The strict definition of these is the direction they are transporting blood in relationship to the heart. Artery away from the heart, vein toward the heart. And then we also have these smaller versions of arteries called arterioles. That suffix, that OLE there, is small. So arterioles, which are basically just going to be our smallest arteries. So they're tiny versions of arteries as they lead into the capillary networks. And then leading out of the capillary networks here, we have tiny versions of veins, so venules. So arterioles are our smallest arteries, venules are our smallest veins, and connecting these arteries to the veins are going to be the capillaries. Okay? And capillaries, this is where the actual exchange is going to take place. We're going to talk about these more at the end after we've talked about the pressure to get things from place to place. Okay, but this is where the actual exchange of nutrients and waste is going to take place in these capillaries. Okay, so let's focus first here on the arteries and the veins. So I'm going to clear off anything we've written so far so it doesn't get confusing. Okay, so arteries and veins both are going to have three layers associated with them. And these are listed here in the middle, and these are these ones here in bold. So the tunica interna, tunica media, and tunica externa. So these are going to be the three layers of both arteries and veins. You'll notice they have lines on both sides showing that they exist in both arteries and veins. Now they might look a little bit different in the two, but the layer exists in both. So we're going to work our way from inner to outer. So the tunica interna, hopefully its name lets you know. So this is the most internal. So this is the one right here on the inside. Okay, lining the very edge. This is where the blood will actually come in contact with. But if you'll notice, again, the walls of these vessels are so thick that we're not exchanging nutrients here. But this is where the blood will come in contact. So because of that, we want a very smooth surface. So this is a smooth, very little friction surface. We want this smooth, frictionless surface here. To help make this as smooth as possible, that tunica interna is lined with squamous. Remember, those are those flat cells, those kind of flattened, squished down cells. Squamous epithelial, because I'm going to have a free surface, right? The hollow space within a over connective tissue. And we want this to be a smooth, frictionless surface so that the blood doesn't slow the blood down. This is actually part of the problem of heart disease, atherosclerosis, is buildup of plaque within these arteries, within these vessels, that then when it builds up here, it narrows the lumen space. The lumen is the opening. And we do need to know that vocabulary term. The lumen is the opening in the middle, and we don't want to do things that would constrict that space. And we want the blood to flow nice and smoothly through that lumen. And so the tunica interna is very similar in both arteries and veins. Like I said, smooth, frictionless squamous epithelium. They both also have this tunica media. Media, middle. So this is our middle layer. This is going to be made of, we'll come over here. So the tunica media is going to contain smooth muscle. So remember, muscle that is under involuntary control. We don't control it. Okay, it's also going to contain elastic and collagen fibers. Basically, it's going to be stretchy so that these vessels, as the blood moves through them, can stretch and come back to their original shape. You'll notice if you look at your artery versus your vein, this is one of our differences between the two, that the artery has a much thicker, smooth muscle layer. Okay, so we'll sum up all those differences in a little bit. So the basic structure, though, is the same: smooth muscle with elastin and collagen fibers as well. This is going to be controlled by the sympathetic nervous system. So some of that flight or fight response is going to occur here. So the sympathetic nervous system is going to control whether these vessels vasoconstrict or vasodilate. So let's break those words down a little bit and talk about what they mean here. Okay, so the vaso part is vessel. Okay, and if they constrict, right, it gets smaller. And if it dilates, it's going to get larger. In particular, we're talking about the diameter, right? So if the diameter of the vessel is normally like this, if I vasoconstrict, it gets smaller. If I vasodilate, it gets bigger. So that can impact blood pressure, okay, and it can impact blood flow from place to place. So in addition to controlling blood flow and regulating blood flow, it will also have an impact on body temperature. Okay, so if we think about the vessels in our skin in particular, okay, if somebody that has, this happens to everybody, but you can see it in people with fair skin, like myself. Okay, if I get really hot, the vessels in my skin, so here's the surface of the skin, the vessels in the skin will vasodilate. And what that does is it brings more blood to the area. So now I have more blood flowing through by the surface of the skin. Blood contains heat because blood is mostly water. So majority of blood is water, right, in the plasma. And water has what we call a very high specific heat, meaning it takes a lot of energy to raise and lower the temperature of water. And so water retains heat very well. So when I vasodilate and have a lot of blood flow by the skin, the heat from the water can start to escape across the skin. And so what that will do is that can help cool me down. So when we are really hot, the vessels in the skin will vasodilate, trying to bring down body temperature. So you're sweating. So you see, again, fair skinned people will look really red in the face, in addition to the sweating. When it's cold, those vessels near the surface of the skin will vasoconstrict so that less blood is flowing towards the surface of the skin, more blood is flowing to the internal organs. And so we're trying to not to radiate as much heat out to the atmosphere because we have less of the blood flow near the surface and more of it near those internal organs that are vital to life. We can unfortunately lose a finger or toe and we won't die. Okay. But if our heart shuts down or our lungs shut down or our intestines shut down because it's too cold, then we're going to have bigger issues. So when it's cold, then the vessels in the skin will vasoconstrict, trying to bring body temperature up, trying to maintain that core body temperature. And so all of that's going to come from that tunica media because that's the muscle control. So arteries and veins both have a tunica media, smooth muscle, elastic and collagen fibers controlled by the sympathetic nervous system. That'll help it determine vasoconstriction and vasodilation, so which can influence body temperature. So the last layer is this outermost layer, this tunica externa. Okay, so it's our outermost layer, and it's going to be made of that tougher, fibrous connective tissue. Basically, we're going to wrap these vessels up nice and tight so they don't burst. So arteries and veins, this is kind of our basic structure of both of these. This tunica interna, tunica media, tunica externa. Okay, so I'm going to clear some of this out, and we're going to look at differences really quickly. Okay, so we looked at the similarities of arteries and veins with their lumen and their 3 different layers. So now let's kind of make sure we point out some of the differences. So hopefully just looking at the picture, maybe you notice some differences. Okay, we mentioned a little bit with this tunica media, the size of the smooth muscle layer in that tunica media is much thicker in an artery than in a vein. We need a little bit more strength in the artery because the pressure is higher. So we need a little bit more of that sturdy muscle there to help with that. Okay, because that layer is thicker, you'll notice the lumen is smaller in an artery, which will bring actually blood pressure up some. Okay, where in the vein, the lumen opening is much wider. Okay, the veins also have valves, which you can see up here at the top of this vein. The veins have valves for the one-way flow, and we'll look at a picture with valves in just a minute. Okay, where the arteries do not have any of those valves. So 3 basic differences between the two. So you'll want to know what an artery, what a vein is, an arteriole, a venule, the different layers of structure between those arteries and veins, the things they have in common, as well as the things that are different. So let's look specifically at valves. These valves work very similar to the valves that we saw in the heart, right? These are one-way valves, trying to keep blood from going in, trying to keep blood going in one direction only. Their structure, again, very similar to how it was in the heart, where as the pressure builds on one side, that will open that valve. But if blood tries to flow back down, it will end up sealing and closing the valve. So there are one-way valves. In addition, so the pressure in an artery is much, much less than it's much higher in an artery than it is in a vein, right? So I go from these arteries that are going to be a little bit bigger, and they're going to narrow down to these arterioles and narrow down even more to a capillary, and then we start getting bigger again. So if we think physics here, right, I've got blood flowing through this big space, so I have a decent, and actually the are much wider than the arteries. So pressure here. Pressure increases as it gets a little bit more narrow, and then the pressure will actually start to decrease as it widens back up as it moves through. So the pressure in the veins is really pretty low. But if we remember, veins are what get the blood back to the heart. So we need to make sure there's enough pressure so that the blood can get all the way back to the heart. So your veins basically have 3 systems built into them to keep blood moving towards the heart. The first one is going to be the valves, preventing that backflow. The second one will be what's called skeletal muscle milking. If you've ever been on a long flight, potentially like an international flight, where you are asked to, you're told that you should either work your calves or stand up, periodically. That's because blood is obviously most likely to pool in our feet because now we're fighting gravity. We have low pressure and we're fighting gravity by trying to get it back up to our heart. So when the calf muscles in particular, when they contract, remember a muscle, when it's relaxed, is nice and straight, but when it contracts, it'll bulge out. So when these calf muscles contract, they squeeze this vein right here. And so that will force blood in both directions, right? But because there's a valve right here at the base, okay, the blood isn't going to go back too far. It has a limited distance. It can go backwards, okay? But that will push blood forward up past the valve in front of it. So between the valves and the skeletal muscle milking, so the moving around, the using of the muscles, that's going to help with our circulation, as well as breathing. Breathing changes the pressure in the abdominal and the thoracic cavities. And basically, what it will do is it creates like a tension type of pressure, a vacuum pulling type of pressure. So when we breathe, right, here's our thoracic cavity, there's your diaphragm there. So when you breathe, that flattens back out. So I get an increase in volume here, a decrease in pressure, right? But my abdominal cavity gets a slight decrease in volume. So pressure is going to go up some there. And so this is when I'm breathing in, right? When I'm taking a breath out, I am decreasing my volume above the diaphragm, which would increase my pressure. Therefore, I'm increasing my volume and decreasing my pressure in the abdominal cavity. So I change the pressure in the abdominal and thoracic cavity. Remember, our heart's up here in our thoracic cavity. So when I breathe in and out and I change these pressures, that helps pull blood back up to the heart. So valves, skeletal muscle milking, and breathing are all things that are helping the veins get the blood returned back to the heart because the blood pressure is pretty low there. Okay, so let's talk about a capillary for a little bit. We're talking about how things move back and forth across the capillary. And we're going to first look at what we call a capillary bed. Okay, so this middle area here, this is a capillary bed. So it's a big network of capillaries. I feel like we have talked about this, but just to be clear, right? If we have an organ, let's just have this nice square organ here. The blood vessels do not go into the organ and then come out of the organ, right? The blood vessels will surround the organ. So they will form a network all over the surface of the organ. So that blood is crossing over the surface of the organ and then and things are moving in and out of the capillaries. But the vessels aren't like a hose that you screwed into the organ, dumping blood straight into the organ. The blood is moving in the capillaries over the surface of the organ, and nutrients are diffusing in and out. What we can't do is have 100%, we cannot have 100% of all capillary beds full at all times. We don't have that kind of pressure, nor do we have that kind of blood volume to maintain that. So at the beginning of each capillary network is what's called a precapillary sphincter. A sphincter is just a circular muscle. Let's define this. So it's just a circular muscle. So the sphincter is relaxed, then it's open. It's not, if it's contracted, then the circle is nice and tight and it is going to be closed. So at the beginning of each of these capillary networks, we have these precapillary sphincters. And those precapillary sphincters, if they are open or the sphincter muscle is relaxed, then blood is flowing through these capillaries. We have the true capillaries right there in the middle. Exchange is taking place here in the middle. If the sphincters are closed, like in this bottom picture, you'll notice I don't have any blood. I'm shunting it, right? So this is the shunt. Right here it's labeled in your top diagram. It's our vascular shunt there. We're shunting the blood just straight through to the other side. Think about when we talked about sympathetic nervous system, for example, and we talked about directing blood flow towards the skeletal muscles so you're more ready for your fight or flight situation, right? So if I'm directing blood to flow towards the muscle, then I have to be redirecting it from somewhere else. So maybe my stomach that's not doing digestion at the moment closes these sphincters at the beginning, shunts that blood through so it can get moved over to the skeletal muscle. So our body will kind of determine where it needs the blood and what needs to happen at that point in time. Okay, so let's take a look at these capillaries. So a capillary is going to be very thin walled, mostly made of squamous cells, usually. Very thin, usually a simple squamous layer. So a single layer that's made of these very thin cells, because we're trying to get nutrients and waste to be able to move back and forth across these. Remember, this is where actual exchange is taking place in these vessels. No exchange is taking place in the arteries or in the veins. So let's look at how we can move materials in and out. So first we have diffusion. We have basically three types of diffusion. If you remember from biology, diffusion is the movement of something from a high concentration to a low concentration. And so it doesn't require any energy, which is beneficial, right? We don't want to spend all of our energy. So we're moving molecules from a high concentration to a low concentration. For example, let's say I had a vessel in systemic circulation. Here's my artery end. It narrows down, forms this capillary network. And here's my vein end. Okay, so when it's coming in on this artery end in systemic circulation, it's going to have a pretty high oxygen, pretty low CO2. So as it moves through this capillary network, oxygen is going to diffuse out to those tissues, and carbon dioxide is going to diffuse from those tissues into that capillary network. So now I end up with a lower oxygen concentration, higher CO2. I can now take that CO2 to the lungs and breathe that off, and I can pick up more oxygen. So that's going to be our diffusion, right? Moving from a high to a low. Something we need to keep in mind. Let's say we have an organ here that, let's just make a square organ. Remember, vessels don't come up in like blood flow into the organ. It literally goes in the organ and comes out the other side of the organ, right? It's not like a tube or a hose that we screw into the organ, pump it full of blood, and the blood comes out the other side. Okay. The blood vessels are going to form these networks around the surface of the organ. So, for instance, in this organ, the capillaries would branch and they just cover the surface of this organ. So the blood is moving, constantly moving through the capillary network, and nutrients are diffusing out, waste products are diffusing in, but that blood is just moving right along across the surface of the organ in these capillaries. So, kind of the three ways that diffusion can happen here is that I can have direct diffusion, which is going right through the cells. Okay, so I'm directly moving through the cells from a high concentration to a low concentration. I also could go through some openings between the cells. Through the intercellular cleft, remember inter is between. So through the intercellular cleft or through these bigger pores, which are also called fenestrations. And those fenestrations are important for things like bulk flow, trying to move lots of large quantities of particular molecules at any point in time across, as well as filtration. Those intercellular clefts could be for, again, molecules that are large, maybe not quite as large as a pore. They're not as good for bulk flow, so they can regulate flow a little bit better. But regardless of the size, whether it's going through the cell directly, through like the kind of medium intercellular cleft or the larger pore, they're still all doing diffusion, where the molecules are flowing from a high concentration to a low concentration. The last way that things could be moving back and forth across this capillary wall would be endocytosis and exocytosis. So again, I know I'm making you go back to biology. Okay, cyto being cell, and endo being inside, and exo being outside. So endocytosis, I'm taking things into the cell. So for example, I have a little molecule here that is in this indentation. That indentation, the membrane circles around it. Now I end up with a molecule inside a vesicle inside of the cell. Or I could have a molecule that's inside a vesicle inside the cell. That vesicle fuses with the outside edge, and it allows the molecules then to be split out of the cell. So exo being out. So endo taking things into the cell, exo taking things out of the cell. So we've got these diffusion, no matter where it's going through, as well as the endo and the exocytosis. So the last thing that we're going to look at here is this diagram down at the bottom. Okay, we are going to end up breaking this into two videos. We'll talk about pressure in our next slightly smaller video. Sorry, this wasn't erasing. All right, so we've got this diagram down here at the bottom. So blood is flowing into the arterial end. So blood is coming in down at this side on the left there into the arterial end and coming out the venous end here on the right. The whole vessel is surrounded by interstitial fluid, which will also be made mostly of water. So as the blood flows into the arterial end, it's going to have a slightly higher blood pressure. You'll notice the arrow width is different between the pressure on the arterial end versus the pressure on the venous end. And so that slightly higher blood pressure is going to be important on that arterial end. The osmotic pressure, you see that on both ends, on both the arterial and the venous end. This is basically the pressure that the interstitial fluid is putting on the outside of the capillary. And so in the beginning, at the arterial end, the blood pressure is high enough to overcome that. And so I have a net pressure out, which will help us get nutrients and fluid out of the capillary. But as the blood continues to move through that capillary, the blood pressure, like we said, goes down. It's still pushing things out, but it's not as strong. And so now the osmotic pressure starts to take over and starts to be stronger. So pushing more things in, which will help push those waste products in as we head out towards the venous system here. Okay, so we'll do some practice work with these different types of vessels, and then we'll be looking at pressure so that we can do our lab.